In this video, we will discuss about tunability of the PA-ECM hydrogel. So far, in our from our previous videos, we have understood that researchers has made a point that this PA-ECM pancreatic cancer cells culture in the PA-ECM hydrogels more uh, will have the characteristics which is similar to the in vivo characteristics that are observed in the patients who are affected with the pancreatic cancer cells. In order to check the tunability, since they have the system where they can customize their ECM. So in order to check the tunability, what they did is they use the PA with the more fibers containing ECM, as the more stiffer environment will generate ECM. So what they find out is that some of the factors that contribute to the ECM, such as Vim and snail ones, are upregulated with increasing the concentration of this PA. However, some of the factors are downregulated, such as post-TN, TIM1, MMP4, and ZEP1 are downregulated. But in the case of pancreatic stem cells, they are upregulated, and the results for their pancreatic stem cells are shown in the supplemental figures. So this indicates that the upregulation and downregulations could depends on the different kind of cells, or it is cell-dependent markers. And after validating that this Vim1 and SNAI2 is upregulated with increase in the PA concentrations, they wanted to check this one of these markers, that is vimentin. They check these vimentin markers with colonies of the vimentin markers with increase in the PA concentrations, and they are able to find out is that this vimentin colonies increases with increase in PA concentrations. And when they check the spare in the presence of this PA plus CaCl2, they find out is that number of spare formations are high in the case of Vim and LOXX2, whereas in the case of Vim, the spare formations are high when the CaCl2 is used, but when there is no CaCl2, the spare formations are low. And CaCl2 is also promotes CaCl2 also promotes this EMT characteristics that can be seen by this increase in the Vim concentrations. And that after validating that their models, PA-ECM hydrogels are able to replicate the results similar to the in vivo in terms of this triggering ECM characteristics in the stiffer environment. When they check the same things for this 2D PCM or 2D monoculture pancreatic cancer cells, what they find out is that they do not see the increase in the markers for the ECM in when the cells are cultured in the 2D mono layers. That can be seen here. Vim is downregulated, ZEP1 is downregulated, and most of them are downregulated, indicating that this 2D culture does not pointing out or indicating that 2D culture does not replicates as close to the in vivo characteristics as been replicated by PA-ECM hydrogels. Thereafter, they did this after validating their tunability using PA. They also want to validate their hydrogel by using this HA, as HA is also found in the literatures to contribute towards this pancreatic cancer cells tumorigenesis. And what they find out is that the closure of the scratch assay does not affect much when they use different concentrations of this HA. That can be seen here, indicating that this HA itself does not cause much impact on this HA itself does not got much impact on this uh, pancreatic cancer cells migrations, as is shown by the scratch closure. And these results are for the 2D monoculture. When they did the same experiment on the 3D the 3D culture pancreatic cancer cells that are developed in the 3D culture, such as PA-ECM hydrogels, they can see that increase in the invasion assays, increase in the invasion properties of the cells with increase in the concentrations, indicating that In the 3D culture, the invasion properties uh, been been replicated as close to the in vivo as in vivo observations, whereas in the 2D culture the invasion properties are not been observed. And similar results they have observed for the spare count. The number of spare count increases with increase in the HA concentrations. And in the literatures it has been uh, it has been mentioned that with this with the combined low and high molecular weight HA can cause low and high molecular weight HA will cause more tumorigenesis or more migration properties because of the some synergetic effect. And when they check their and when they check these observations in their cells that are grown under this PA-ECM hydrogel with only collagen, they see that the tumor formations are low, or tumor volume growth is low, whereas when they use this collagen and HA combined, they see that tumor growth formations is high, indicating that these observations are this literature hypothesis is also been replicated in their model. These previous literature findings can also be replicated in this model, indicating that their models is much more efficient than the previously existing models for the pancreatic cancer cells studies. So I hope you understood how they have presented the tunability of the PA-ECM hydrogels towards pancreatic cancer cell culture. If you want to understand more about their experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.